Hello guys and welcome back to Excelsior Academy. So we are doing cytoskeleton and the cell movements. In the previous lecture, we saw like how active filaments are helping, how they are polymerize and depolymerize. Now the same way, these active filaments are helping in the cell movement. And to have cell movement, cell move करे उसके लिए they start forming the protrusions. Okay, so in the last lecture we saw like how in the microvilli it is present and how they are moving with the help of the myosin, the actin motor protein which is present, right? In the same way, now let's talk about how exactly the entire cell will move. Okay, now so in contrast to microvilli, there are many surface protrusions are there which are formed in response to the environmental stimuli and are involved in the cell movement. So we know whenever there is a stimulus and cell needs to move, whether it is to reach the wound area or where the nourishment is available, the cell is going to move towards that, right? And for all this, you need these active filaments moving forward. So the movement of cell across the surface, it is a basic form of cell locomotion, which is undertaken by many of the cells. For example, like movement of the amoeba, the migration of the embryonic cells during the development. Now about the WBCs to fight the infection, that is to reach the site of the wound healing, and also the spread of the cancer cells. The similar types of movement are also responsible for the phagocytosis and for the extension of your nerve cell process during the development of the nervous system. So all these movements, okay, they all are nothing but the local extensions of the plasma membrane, which is done by the actin filaments. Okay, so here is the example, like this is a where macrophages is eating the engulfing the tumor cells, and then here the amoeba which is trying to move, and here the fibroblast which is making the protrusions over here. Okay, now this is just one example where the WBCs are reaching the site. For example, it starts making over here the protrusions. Can you see these small single lines protrusions are there? So these are first formed and then they try to squeeze themselves between the two endothelial cells. Okay, then they start coming basolateral leading edge. So this is known as the leading edge. Normal, then usually they will followed by the nuclear lobe formation and the insertion. पूरा का पूरा nucleus आपका आएगा, उसके follow करता हुआ पूरा cell आएगा, ठीक है? Then they start squeezing, यानी कि क्या? So you have broken the connections between the two endothelial cells. So as I said, in between the two cells, okay, between the two cells here you have got many tight junctions are present. So those junctions are being removed, and then the cell, the cell starts squeezing itself. And then it will start coming slowly, these tight junctions will be resumed and the cell will reach its site. Okay, so this is how any defensive cell, they reach their wound healing site. Now, pseudopodia. What is pseudopodia? So pseudopodia are the extensions of the moderate width based on the actin filaments cross-linked into a 3D network, which is responsible for phagocytosis and for the movement of amoebas across the surface. So here what you see is wherever these kinds of projections are formed, these are your pseudopodia, जिसका कोई shape नहीं है, ठीक है? This is pseudopodia. Now, laminopodia. So they are broad, sheet-like extension at the leading edge of the fibroblast, which contain network of actin filaments. So this also you have seen in the previous lecture where I was talking about the fibroblast. So like there's a narrow sheet-like projections were there, which was helping the fibroblast to move. Okay, so those are your laminopodia. Then coming to the filopodia, they are the very thin projections of the plasma membrane supported by actin bundles and they extend from the laminopodia. यानी कि क्या? कोई सेल है लैमिनोपोडिया बना है, वहां से आपका फिलोपोडिया बनेगा पतले पतले से, ठीक है? I'll show the figure, it will be more clear. Now the formation and the retraction of these structures during cell movement is based on the regulated assembly and disassembly of active filaments. So in short, it is all about the polymerization and depolymerization of the actin filaments. Okay, here you see, ये आपका laminopodia, the broad way when it is trying to move, and then if you have got this thin projection, then that is your filopodia. Okay, now coming to the cell movement. So the cell movement. So cell movement, or it is the extension of long cellular process which involves coordinated series of movements, okay, which can be viewed in several stages. First, so what is happening? First, the protrusions like pseudopodia, laminopodia, filopodia must be extended to establish a leading edge of the cell. What does it mean? What does this mean? It means if you have a cell like this, okay, first, whether it is forming a pseudopodia, laminopodia, filopodia, it needs to form a leading edge like this. उसको पता होना चाहिए कि वो किस तरफ जा रहा है, ठीक है? So they should know the direction. So the initial movements of most of the cells are mediated by the extensions of the filopodia. Okay, so जो एक आपका laminopodia, let us say that this particular cell is there, it needs to move in this direction, so it is going to form something like this. Okay, so this forms your laminopodia. Ab isme bhi, if you get thin like projection, that becomes a filopodia. Okay, so these extensions must then attach to the substratum across which the cell is moving. So it means all this, in short, is helping your cell to move towards the target area. Did you get this point, guys? Look it over here. So in this figure, see, you can see, though these interactions are made as we saw in the previous lectures, right? Integrins, they make the interactions between the cell and the extracellular surface, right? Now it forms the extension of the leading edge. So now the cell knows that it has to move in this direction. So it is making, and when it is making the leading edge, new interactions will be made here. So these are the new interactions made. And retraction of the trailing edge. What does it mean? अब उसको आगे बढ़ना है, तो obviously when it is making new interactions, in the back, it is going to leave the previous interactions, correct? So here they will start losing this connections, and here they will start making new connections. And eventually what will happen? More surface, it will come like this, and here it will become like this. So इस तरह से क्या होगा? आपका cell आगे की तरफ move हो रहा है. Okay, did everyone got this point? Yes, Akshay, is it clear to you?
Yes, ma'am.
Yeah, okay. This is just a video, please, okay? Like, please observe this video and let's see like how much the understanding is coming.
The shape of the cell is highly polarized.
Let me share the volume over here.
The shape of the cell is highly polarized. A broad, thin veil of cytoplasm called the lamellipod is at the front of the cell, and the back of the cell is tapered into a thin tail. The nucleus and major organelles are located in the cell body. A network of actin filaments fills the entire cell, while contractile myosin molecules are located in the cell body. Cell crawling takes place in 4 stages. In the extension stage, the cell pushes the lamellipod forward. This movement is driven by rapid polymerization of actin filaments at the leading edge of the cell membrane. In the next stage, the lamellipod adheres to the underlying surface using specialized membrane adhesion sites. Firmly anchored, the cell is now ready to generate the force to move the cell body forward. The third stage is translocation of the cell body. This is accomplished by contraction of actin and myosin bundles at the lamellipod cell body border. After the cell body translocates, the back of the cell releases from adhesion sites and moves forward.
Okay.
The shape of the cell is...
Everyone got it? Any doubt so far?
Yes, ma'am.
I mean, there is no doubt, right? Okay. So now in most cases, the cells will move in response to the signals from the other cells or the surrounding, right? For example, in the case of the wound healing, the cells are present at the edge of a cut. It moves across the extracellular matrix to cover the wound. So just now, like in the previous we saw, right? So these formation of the cell surface protrusion in response to extracellular stimulus is regulated by, again, small GTP binding proteins known as Rho. So already we have talked about RAF and RAS. to the same family, this row is also belonging. Okay, do not get confused with this row protein and the other termination factors. Okay, these are different. So signals that stimulate cell movement activate receptors in the plasma membrane, leading to activation of row family. Now, this row family members will activate or promote polymerization by stimulating your ARP2/3 complex. So what does ARP2/3 complex does? It makes or it creates branches in the actin filaments. If you remember, so profilin is bringing your ATP-G actin to the plus end. Cofilin is breaking breaking your actin filaments, creating new sites for polymerization and depolymerization, whereas ARP2/3, it's actually helping in creation of the new branch. Okay. So this gets activated with the help of the row family members. Now, the row family members also activate formins, which extend the branch filaments initiated by the ARP2/3 complex. Now, cofilin is also playing an important role in remodeling. So they know that when they cleave the existing filaments, they provide new plus end and the minus end. The growing actin filaments push against the plasma membranes and drive the formation of cell surface protrusions. Okay, so I hope this is clear. Now, as a new actin filaments extend into the growing cell process, they provide pathways for the delivery of vesicles containing lipids, proteins needed for the continued extension. So if you remember in the video where I have shown how this motor proteins like microtubule motor proteins, kinesin and dynein, right? So they carry the vesicles. So if you have seen clearly, you must have seen that along with the microtubules, the actin filaments are also present. So they are just forming the pathways. As I said, like the analogy I gave where I said the microtubules act like a national highways and actin filaments act like a local paths, as a kacha rasta type. Okay? So whenever you do any part of your cell bio, okay, whatever theory we are doing, just go back and watch that video again. So that becomes more clear then. Your picturization becomes more clear. Okay, so the cell attachment to the surface, as we know, is known as the focal adhesion. Remember, we spoke about the focal adhesions. Now, the final stage of cell migration, retraction of the trailing edge. It is again the activity of small GTP binding proteins of ARF and the row families, which regulate the breakdown of the existing focal adhesion. So, इससे क्या होगा? आगे आपके new attachment बनते रहेंगे. Something like this it was, right? So they are making new focal adhesions जो पीछे है, that they are going to leave, so that the cell is moving in this direction. Okay, so the stress fibers which is connected to the new focal adhesions at the leading edge then pull the trailing edge of the cell forward, and hence now the cell is moving. Okay, now look at over here. So when in the video when they were showing that at the lamellopodia, new actin filaments were forming, right? Like this, they show like this new actin filaments are forming, right? So how exactly this is happening is, now look at this. This is the plasma membrane. This is your surface. Okay. So what is happening? We have got the row family proteins activating your ARP2/3 and formin. Formin क्या होता है? जहाँ पे आपने शुरू किया है. If you remember the formin dimer where the nucleation starts, right? So these row proteins are actually helping in initiating the branches of the actin filaments. Profilin helps in binding of the G-actin bound to ATP. Cofilin, we know that it removes the or makes a cut in the actin filament. Okay. Now what is happening? So this is how these row family proteins are helping in starting of your actin filaments. समझा? See, कोई भी चीज बनने के लिए you need the base, and that base is provided by the row family, and this row family is gonna activate your formin. तो formin dimer में आपका 3 G actin आता है, जहाँ से nucleation शुरू होता है, and then they keep on adding the monomers in order to form the filamentous microfilaments. Okay, I guess this is another video I have added for the better understanding. Let's watch this. So guys, before I start the video, any doubts, like are you able to get like how the cell are moving, how these proteins like profilin, cofilin, formins, R2/3, R row family, all these proteins, how they're helping in the formation of the protrusion so that the cell can move? Is the concept is clear? Yes, Krishita? Shruti?
Yes, ma'am.
Okay, let's watch then. Bapre, sound is too much.
Movement. It is essential to the function of animal cells. A type of immune cell, these lymphocytes might
Can you see these thin projections? These are your filopodia, okay?
migrate toward inflammatory signals as part of immune surveillance and defense. You see, the plasma membrane and the cell shape are constantly changing in order to achieve cell motility. How does this happen? Like a scaffold, this network of cytoskeletal fibers connects every corner of the cell and gives a cell its shape. Changes in cell shape rely heavily on one type of cytoskeletal protein, known as actin. The actin cytoskeleton is highly dynamic, and its filaments continuously assemble and disassemble throughout the moving cell. The building block of the actin cytoskeleton is a small globular protein called G-actin, which is normally bound to ATP as a monomer. These ATP-bound G-actin monomers assemble, or polymerize, into filaments, known as F-actin, over 3 phases, called nucleation, elongation, and steady state. Nucleation occurs as 3 ATP-bound G-actin monomers form trimers. Then rapid linear polymerization occurs in a process called elongation. G-actin can bind reversibly to either end of the growing filament, known as the barbed or plus end, and the pointed or minus end, but monomers preferentially bind to the barbed end. Bound G-actin monomers undergo ATP hydrolysis, producing an increase of ADP-bound G-actins at the pointed end of the filament. These G-actins dissociate more readily than the ATP-bound G-actins at the barbed end of the filament. Thus, actin filaments undergo treadmilling. As the reaction reaches a steady state, G-actin monomers are constantly added to the barbed end and removed from the pointed end of the filament. At the leading edge of a moving cell, actin filaments are continuously treadmilling, branching off, and cross-linking. These activities push the plasma membrane, forming cell protrusions, such as wide, blunt projections called lamellopodia, and narrow, pointed projections called filopodia. Filopodia are formed by rapid elongation of parallel actin bundles, regulated by elongation factors such as formin. Small cross-linking proteins such as alpha-actinin bind to these filaments and strengthen these parallel actin bundles. In lamellopodia, an actin-binding protein complex, ARP23, binds near the leading end of newly formed actin filaments and nucleates branching. Large cross-linking proteins, such as filamin, bind to these actin filaments, and support the structure of this branched network. This continuous branching off and cross-linking strengthens the protrusive force needed to overcome the compressive force of the plasma membrane. As the cell moves forward, adhesions occur along the leading edge of the cell as actin bundles anchor to the surface via adhesion molecules. These newly formed adhesions are connected to old adhesions via contractile stress fibers, consisting of myosin filaments and antiparallel actin bundles. The cell is pulled forward mainly by contractile forces generated by myosin acting on the actin filaments. As the contractile fibers contract, the rear of the cell comes forward. These dynamic changes in the actin cytoskeleton occur in a fraction of a second and work in concert to facilitate the continuous cycles of cell protrusion, adhesion, and contraction. This enables cell motility, ultimately allowing lymphocytes to perform their duty of immune surveillance and defense.
Okay. So it was just like the summary as well as like what just now I have taken. So that's a really very beautiful video which is covering entire points. So any doubts? Tanvi, was it clear?
Ma'am had mentioned contractile forces. I didn't get that.
Okay, so they are also contractile stress fibers. So they are also type of the stress fiber. So if you remember, there was one figure in which I said that there are different types of the stress fibers. Transverse tha, dorsal tha, ventral tha, right? So when they said about the contractile, what they are saying is that below, okay, ऐसा कुछ दिखा रहे थे cell, right? So what they are saying is that जैसे जैसे सेल आगे बढ़ रहा है न्यू एडिशन साइट्स दे आर मेकिंग राइट एंड यहाँ पे उसको पीछे वाले छोड़ने हैं फॉर विच जैसे हम आगे बढ़ते व्हेन वी वॉक ऑन द रोड व्हाट वी आर डूइंग बिकॉज ऑफ द फ्रिक्शनल फोर्स वी पुश द रोड towards the back and then we move ahead, right? Over here also these are the contractile stress fibers. ऐसा रहेगा. So they have got the contacts over here and here. Okay, so when they make a new addition site over here, new contact has been made here. Now myosin, what myosin is going to do? It is going to help in the contraction. So वो क्या करेगा? Actin filaments को ऐसे सिकुड़ेगा, अपनी तरफ ऐसे लाएगा, ठीक है? So what will happen? The contacts will be broken over here और यहाँ पे आ जाएगा. So what had happened? Cell यहाँ से यहाँ तक move हो गया. राइट और अब जाके एक और नया एडिशन साइट बनाएगा. अगेन द मायोसिन विच इज प्रेजेंट ओवर हियर इट इज गोइंग टू कॉन्ट्रैक्ट देन new addition point is made. So this is how the cell is moving, like below what we were not able to see, the addition sites. Okay, so new addition sites are made and the previous ones are broken. Okay, so this is happening because of the contractile stress fibers, again actin only. Now how myosin is helping in the contraction, we'll talk about it when we discuss about the sliding filament theory. I hope everyone must have heard about it, sliding filament theory. During the muscle contractions, usually we talk about the sliding filament theory. The same phenomena is used whenever the actin filaments are there and myosin is contracting so that the cell can move in the forward direction. Clear now, Tanvi? Abhi theek hai?
Yeah, ma'am.
Okay. So this is, yes.
What's the difference between laminopodia and filopodia, ma'am?
So that's what they showed you, right? In the video also beautifully they showed. If suppose this is the portion which is moving, this is laminopodia, where you have got the broad actin network. Okay. Now, if it is making a thin like projections, which you saw many as a, it was like thorn on the surface of the cell which was moving. These are filopodia.
Okay.
Okay. Yeah. So if you ask like why filopodia and why laminopodia, so what happens is that when a cell is moving, so these filopodia is also acting like an antenna. like in the surrounding, if suppose any chemical signal is floating here and there, then they should be able to detect, because even though it is a filopodia, inside to आपका actin filament है, but ऊपर तो receptors है ना. So if suppose any chemical molecule is present, which is a danger or which is a food, whatever it is, so they may detect over here. And they will understand whether I should move in this direction या पीछे की तरफ भागे. Okay.
Okay, ma'am.
This is another video, like when you tag it with the fluorescence. So see, here it is forming the laminopodia, and these finger like very thin projections are the filopodia, and the entire cell is moving. So when you do in future the research, so this is how you actually understand. This is like real live imaging, real cell, sorry, I mean to say alive cell and real imaging. So when the cell is in the progress, it is alive, and then you tag it with the dye, right? Fluorescent dye, then you can see how it is, actually moving. Now coming to the myosin motors. So every time we were saying that myosin is responsible for the contraction, right? So let's talk about myosin motor, how exactly they are helping in the movement. So without myosin, none of the cell can move anywhere. So myosin are really very crucial. Okay, they are responsible for all types of movements. So it's a molecular motor protein. It converts chemical energy in the form of ATP to mechanical energy, thus generating a force and a movement. So each movement of the myosin needs ATP. For example, let's talk about in the case of the muscle contraction. So interactions of actin and myosin are responsible not only for the muscle contraction, but all types of movements which is happening in the non-muscle cells also. For example, in the cell division and the intracellular transport of vesicles and the organelles. So abhi hum aage dekhenge like how in the cell division when the division is happening between the two. Okay, I mean to say when ऐसे cell जब आपका divide होता है, so we say that right from here it is going to get divide. So here it is going to form the contractile ring. That ring is again made up of actin filaments. ठीक है? Yeah. So let's talk about the muscle contraction. So muscles are highly specialized for the single task axis contraction. There are three different types of muscle. Skeletal muscle responsible for all voluntary movements, cardiac, which pumps blood from the heart, and the smooth muscle, which is responsible for involuntary movements of the organs such as stomach, intestine, uterus, blood vessels. Now, in both the skeletal and the cardiac muscle, the contractile elements of the cytoskeleton are present in highly organized array. Okay, so it's not that they're randomly present, they're very, very organized, and that's why when you see the muscle cell, okay, each fiber, when you take myofibrils, we say, when you take each myofibril, you can see that this organization results in the striation. इसका मतलब क्या, कहीं कहीं पे dark होगा, कहीं कहीं पे light होगा. Okay, so these skeletal muscles are the bundles of the muscle fibers, which are around 50 micrometer in the diameter. They are formed by the fusion of many individual cells during the development, and they consist of myofibrils. So I'll show how they are looking. So each myofibril is organized as a chain of contractile units called sarcomere. यानी कि एक sarcomere जब contract होता है, there is a movement. So for the entire muscle, several of sarcomeres, when they are getting contracted, then only the action is taken by the hand or wherever the muscle movement is required. Okay, look at over here. So here they have taken the muscle. So muscle is made up of bundle of muscle fibers. Each muscle fiber is then again having, so many cells together. So then they have got the plasma membrane, then it is having the myofibril, and what do we see is that one each sarcomere. So sarcomere matlab kya hua? The unit between the two Z disc. Okay, now the dark ones are the A band, the light ones are the I band, we say. Okay, this is how it looks, I band and the A band. Now the same thing is given over here. So दो Z disc के बीच में एक sarcomere हुआ आपका, ठीक है? Now look at over here. So A band is having your first, the middle one is the middle line, M line. Of that, both the sides, you see that myosin is present. Okay, and to both the sides, up upper nature, you can see that the actin filaments are present. Can you see? These are your actin filaments. And they are present in an array, यानी कि totally organized way में present है, right? Now हर एक actin filament के बीच में एक myosin present है. I mean, it's not one actually, they are the collections of myosin. This one particular projection जो ऐसे करके दिख रहा है, one, this is one myosin. So here you can see so many are present. Okay, now when it is contracting, it means what? There is a movement, it makes an interaction with this and pulls this side. This one is going to pull this side. So you see that the direction of myosin दोनों का opposite way में जा रहा है, right? Now when they make interaction with the active filaments, they are going to push in this direction. ये इसको यहाँ push करेगा, and that's how the contraction is taking place. Okay, I hope everyone got little idea. It's okay, we will be doing this in detail. And yes. So now here each contractile unit, which is also known as sarcomere. So the ends of each sarcomere are defined by the Z disc, and each sarcomere is going to have the dark bands and the light bands. So dark bands are called the A because they are anisotropic when viewed under the polarized light. So इससे वो dark दिखेगा, ठीक है? And then light bands are there for your isotropic. Now the bands correspond to the presence or absence of the myosin filaments. So अगर आप ध्यान दो, हमने अभी देखा I band में myosin is not present, whereas the thicker filaments of the myosin are present in the A bands. Okay, let me just go back and show you. Okay, so here, see, this region is going to be darker. A band, जो आपका dark है, it is because of the presence of your myosin. And whereas on the I band, it is lighter since there is the absence of the myosin. Okay. So the actin and the myosin filaments overlap in the peripheral regions of the A band, whereas the middle region contains only the myosin. The actin filaments are attached at the plus ends of the Z disc. The myosin filaments are anchored at the M line in the middle of the sarcomere. Let's go slowly because there are so many proteins which is going to be involved in this. Okay, is this much is clear? The A band, I band, what is the H zone? If you see this is the H zone, M line, it means only myosin is present. Is this much is clear to everyone? And here the plus and the minus end is about the actin. Okay, so it's about the actin. Okay, now the sliding filament theory. So this theory is telling like how myosin is helping in the contraction. So during each muscle contraction, each sarcomere shortens, which will bring the Z disc together. So there is no change in the width of the A band, but both the I bands and the H zone almost disappear. So these changes are explained by the actin and the myosin filaments sliding past to one another so that the actin filaments move into the A band and the H zone. So this is just like myosin holding the actin filaments in both the direction and pulling towards the center. And that's how the contraction is happening. Okay. Now the muscle contraction thus results from an interaction between the actin and the myofilament that generates their movement. And the molecular basis kya hai? The binding of myosin to the actin filaments. Now this is really very, very crucial, like how myosin is binding to the actin. So before we move ahead, let's know what is myosin. So in the case of actin myosin, okay, actin filament, it is the myosin 2. We have got different types of myosin also. There are like different versions of myosin. So whenever we talk about contraction, please remember it is myosin 2. Okay, so it's a very large protein. It has got 2 identical heavy chains and 2 pairs of light chain. Each heavy chain consists of globular head region with the long alpha helical tail. Head region and the tail region is the head region जो रहता है वो आपका actin filaments में bind करता है. ठीक है. Now the alpha helical tails of the 2 heavy chain twist around each other in a coil structure to form a dimer. And the 2 light chains associate with the neck of each head region to form the complete myosin 2 molecule. Like this is how. So you have got this head regions are there, and these are the coiled of the 2 alpha helical tails. So these are the globular head region which makes interactions with the actin filament. Okay. Now, the thick filaments of muscles, they have got so many myosin presence over there. Now look at the figure. So here, as I said, ये आपका रहता है plus end, ये रहता है आपका minus end, your actin filaments. Okay. Now these are nothing but so many cluster of myosin presence. You see, did you notice this one? Now each myosin head is making interactions with your actin filaments. Do you see over here? Both sides, ऊपर नीचे दोनों की तरफ कर रहा है. And this myosin molecules, myosin proteins are arranged on another protein called titin. Okay. Now to have this rotation of the actin filaments, see कैसा होता है जब आपका myosin interact कर रहा है with the actin filaments, it means there is a movement which is happening in the actin. ऐसे twist होता है. Now that twist is brought about by the nebulin. Okay. So far so good, guys. Are you able to follow? So the globular heads of the myosin, they will bind to this actin. They're forming the cross bridges between the thick and the thin filaments. Okay, so far so good. Okay, now here the orientation of the myosin molecule is important. Let's see how this is happening. So the polarity of actin filaments similarly reverses at the M line. Now, what do you mean by reverses? See, यहाँ पे अगर आप फिगर में देखोगे, so you see that यहाँ पे आपका myosin filament का ये direction है. यहाँ पे क्या है? दूसरा direction. It becomes like this, right? So that actin filaments will move this side. वो इसको पकड़ के इस तरफ खींचेगा, ये इसको पकड़ के इस तरफ खींचेगा, and hence Z disc आपका जो है short हो जाएगा, resulting into the contraction. Okay. Now contraction results from the movement of the myosin head groups along the actin filament in the direction of the plus end. The movement slides, this movement slides the actin filaments from both sides of the sarcomere towards the M line. So what is happening? Towards the M line. So because it is moving towards the M line, SA and SA. So the entire thing from here to here, it will become this much. Okay. Guys, are you able to follow? That's why I'm going little slow. Are you able to follow? Everyone? Shruti?
Yes, ma'am.
Okay. Now, the activity of myosin. Now, activity of myosin is it's a molecular motor, and I said for each step it needs ATP. Okay, so this is mediated by the ATP binding. So ATP hydrolysis derives the repeated cycles of interactions between the myosin head and the actin. So ये जो भी chemical energy है ATP की, on hydrolysis it is helping in changing the shape. हमें पता है हमेशा से whenever ATP hydrolysis is happening, always there is a conformational change, right? Or any structural change is there of the protein. So during each cycle, conformational change in myosin results in the movement of myosin head along the actin filaments, which results in the shortening of the actin filaments. Okay, let's see how. So first, let's go with the help of the figure. And to make it more clear, I have added a video also for your better understanding. Okay, yeah, let's see. So here, this is in the one direction हम दिखा रहे हैं, ठीक है? तो same way आपका इस direction में भी होगा. So you have got the plus end and the minus end of your actin filaments, and the myosin is making interactions with the actin filaments over here. Can you see it? ये यहाँ पे इसने इसको bind किया है, ठीक है? So what happens is that this nebulin protein, they actually slide or they actually twist the actin filaments, जहाँ पे पहले accessible नहीं रहेगा और बाद में accessible होता है. ठीक है. Now it is a dissociation of actin myosin complex when ATP is being added. तो पहले क्या है? In the absence of ATP, it is making an interactions with the actin filaments. अभी आपने ATP bind किया है, ठीक है? Now ATP hydrolysis is there. This results in the conformational change, which will result in the movement of the myosin head. So अभी क्या हुआ? ये पहले उसका सर ऐसे था, प्रोटीन का, मायोसिन का. अब क्या हो गया है? अब वो ऐसे हो गया है, ठीक है? तो मायोसिन हेड binds to the new position on the actin. So पहले उसने यहाँ bind किया था when it was having this direction. This direction. Now with the ATP and ATP hydrolysis, the direction has changed. वो ऐसे बन गया. Now it is making connections with another portion. जैसे कि यहाँ से, sorry यहाँ पे था. Now it is making to the next actin region. Can you see the difference? They have shown in the difference in the yellow region. So if you see carefully, पहले यहाँ पे था उसका association. Now they have made to the new one. right? Now the myosin head returns to its original positions. तो अब क्या हुआ, पहले उसने ऐसे बाइंड किया, जब वो ओरिजिनल पोजीशन में आया, to come back to this position, by the time actin was slided in this direction. Guys, did you get it? Really very beautiful phenomena happening inside our cell and very regulated. Yes, anyone who was not able to understand? Tanvi, Akshay, is it clear?
Ma'am, can you repeat again?
Yeah, sure, sure. Because this is really very important. I really want everyone to understand this thoroughly because कैसा होता है ना, इसके action से ही base question आएगा. Okay. So this much is clear, like you have got the actin filament, plus end and minus end is there. Then we have got the myosin. So instead of showing these many bundles, I'll just show only this portion. Okay. Now, what is happening here? Oh, 1 second. So I'm saying that here, this is how your myosin head is present. Okay. Now it is present, let us say 1, 2, 3, and 4, 5, 6. These are the 6 actin regions where the myosin head can interact. So currently it is interacting with the 4th one. ATP comes and binds. So when ATP is binding, the interaction between the actin and the myosin is broken. Okay. Or we can say that it got dissociated. Now ATP hydrolysis is bound to your, oh, so sorry. Now ATP is bound to your myosin, right? So this results in the dissociation of actin and myosin interaction. So what will happen later? ATP hydrolysis happens. ATP hydrolysis हो रहा है तो आपका ये जो shape है myosin का ऐसे हो जाएगा, different way, ठीक है, दूसरा way में हो जाएगा. I am so bad in drawing. And especially, you know, using this notepad. Something like this. Okay. Now, when it is changed, okay, this will go and bind to your third region, third side where it is interacting with the actin. ठीक है, ये open होके इसने यहाँ पे bind किया ऐसे. Now, what is happening is that it has to come back to its original position. So पहले क्या हुआ, जब इसने third side पे interact किया, PI release हो गया. Now it is changing its shape again back to this shape, back to this shape. So जब उसने वो किया, इतने में उसने actin को यहाँ push किया. So it's not the myosin which is moving, it is the actin which is moving. Okay, so when it is changing its shape, so it has to push the actin in this direction so that when from here to here, this shape it makes, this portion needs to interact with this region. तो ये करने के लिए उसको actin को इस तरह push करना पड़ेगा, ठीक है? And eventually the ADP is getting released. So what is happening now? अगर आप starting से देखो, यहाँ पे this light yellow color they have shown first interaction. Later they are showing the next, which is next to it, right? So now it has reached over here. So all these myosins, okay, ये सब एक दूसरे को ऐसे से bind करते हैं and totally they are helping in the movement. And now since actin is moving in that direction, it is known as the actin ka sliding filament theory. Okay, now it is clear. Let's see the video also. Maybe that time it will be more clear. Yeah. So before that, as I said, there are many proteins which needs to be remembered so that this can be done. 1 second. Yeah. Okay. So what are these proteins? So if you remember, I was talking about the nebulin, which is helping in the twisting of the actin filaments, right? So all these proteins are present over here. So ये कैसे करते हैं? You have got those tropomyosin. The nebulin is nothing but the tropomyosin, जिसको ऐसे wrap करके रखा हुआ है. If you see, these actin filaments are wrapped by the tropomyosin. Now we have got troponin complex. Okay, there is this troponin complex. Troponin I, C, and T is there. Okay, so because of this, what has happened is that your tropomyosin is blocking all the myosin binding site. As soon as calcium is released, it goes and binds to this troponin complex, what is happening? There is again the twisting of the tropomyosin, which is now releasing your myosin binding sites. Did you get it? See, another figure. So you have got this troponin complex, I, C, and T is there. And in the actin, this is a region where myosin binding site is there, which is blocked by the tropomyosin. Now, once calcium comes and binds to the troponin complex, what is happening? The tropomyosin is moved and the myosin site is exposed, to which the myosin head is going to bind. Okay. Is it clear?
Yes, ma'am.
Okay, let's watch the video and then you can, just understand and correlate like whether your understanding and the video is clear or not. Okay, just compare yourself.
Muscle contraction. In this module,
Sorry, the sound is audible to you? The voice is coming? The video which you're watching, whether the audio is coming, are you able to hear?
The volume is
The volume is?
very low.
Okay, okay.
You will learn about the mechanism.
Now it is clear?
Yes, ma'am.
Okay.
Muscle contraction. In this module, you will learn about the mechanism of muscle contraction. All our body movements, such as walking and running, are possible due to the presence of muscles inside the body. You already know that myofibril, that is, the simplest unit of muscle, contains contractile proteins that help in the contraction of muscles and thus in body movements. The sliding filament theory is the most accepted theory, which best explains how these proteins work to cause contraction of the muscles. The theory states that contraction of a muscle takes place by the sliding of the thin filaments over the thick filaments. Let us understand the steps given in the theory. The process of muscle contraction begins by a signal from the central nervous system, CNS. The signal is brought by the motor neurons from the CNS. These motor neurons are connected to the muscle fibers. The motor neuron with which a muscle fiber connects is called a motor unit. The junction between the motor neuron and the sarcolemma of the muscle fiber is known as the neuromuscular junction or the motor end plate. This junction is actually a synapse, that is, a small gap between the neuron and the muscle cell. The signals from the CNS are transported as electrical impulses, also known as action potential, across the synapse. A neural signal, on reaching the neuromuscular junction, releases a neurotransmitter, acetylcholine, thus generating an action potential in the sarcolemma. This action potential spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm from the sarcoplasmic reticulum. This in turn increases the level of calcium ions in the sarcoplasm. The increased level of calcium ions leads to the binding of calcium with the subunit of troponin on the actin filaments. This interaction triggers a conformational change in the tropomyosin, thus preventing it from masking the active sites for myosin present on the actin filaments, leaving them exposed. With the active sites of actin now exposed, the myosin heads start a binding process with actin, thus forming a cross bridge. The energy needed for the cross bridge formation comes from the hydrolysis of ATP attached to the myosin into ADP and inorganic phosphate. The attached actin filaments are therefore pulled towards the center of the A band, bringing the Z lines closer to each other. This ultimately results in the shortening of the sarcomere, that is basically the contraction of the muscle. You can clearly see that there is a reduction of I band during contraction, while the A band remains of the same length. ADP and inorganic phosphate are released during the shortening, thus binding the myosin head to the actin tightly. The myosin remains attached to the actin until a new ATP molecule binds to the myosin head and the cross bridge is broken. The ATP is again hydrolyzed by the ATPase found on the myosin head, and the cycle of the cross bridge formation and breakage is repeated, causing further sliding. The cross bridge cycling sequence continues to repeat till the muscle fiber is excited by the signal from the CNS. As the signal stops, no further release of calcium ions from the sarcoplasmic reticulum occurs. This enables the pumping back of calcium ions from the sarcoplasm back into the sarcoplasmic reticulum. This pumping back of calcium ions results in the masking of active sites on the actin filaments. This in turn causes the return of Z lines back to their original position, that is, relaxation. All the muscle fibers do not respond to the signal in the same interval of time, that is, the reaction time. The interval between the signal and the beginning of a response is not constant in all the muscle fibers. The slow twitch muscle fiber contracts slowly and utilizes oxygen better. What makes it slow is the components present in it. First is the myoglobin protein. It is an iron-oxygen binding protein found in muscle fibers. It is similar to the hemoglobin present in red blood cells. Myoglobin content is very high in slow-twitch muscle fibers. Thus, these muscles are also called red fibers. The second component is mitochondria. These muscles also contain a large number of mitochondria, which can use the oxygen stored in them for ATP production. These muscles, therefore, are also known as the aerobic muscles. For example, extensor muscle on the human back. The fast twitch muscle fibers, on the other hand, contract quickly and make use of the anaerobic metabolism. They generally contain fewer myoglobins, thus are pale or whitish, hence also known as white fibers. The number of mitochondria are also few in them, hence they have a low oxidative capacity. That is, the ability to use oxygen is low. The amount of sarcoplasmic reticulum is high in white fibers. For example, the muscles of the eyeball. Let's recap. The sliding filament theory is the most accepted theory, which best explains the mechanism of muscle contraction. A motor neuron carries signal from the CNS to the muscle fiber, which generates an action potential. The action potential causes the release of calcium ions from the sarcoplasmic reticulum. Calcium ions bind to troponin on actin, thus exposing the binding sites for myosin. The myosin heads bind to the actin, forming a cross bridge that pulls the actin filaments, causing them to slide over the myosin filaments, thus causing contraction. Once the motor neuron stops sending the signals, calcium ions are returned back to the sarcoplasmic reticulum. This inactivates the actin, cross bridges are broken, and the muscles relax. Muscle fibers are classified as red muscle fibers and white muscle fibers, depending on the amount of red-colored myoglobin in them.
Okay, that's all, guys. Any doubts, any area? So this video also gave additional information regarding the myoglobin and the red and white muscles. That's okay. Any doubts regarding the sliding filament theory, like how the actin and myosin together are playing important role in carrying out the contraction? Anywhere, any point which was not clear? Okay, no doubts are coming. So I guess I'll take that as everything is clear. Okay, so I'll be stopping here now, and let's meet tomorrow morning, and we'll be starting with the microtubules. So here actin and the motor proteins are over for actin. So we know that there are 3, right? So actin is over, and next we'll be talking about the microtubules, and last we'll talk about the intermediate filaments. Okay. With that, this topic is over, and there is another topic called cell adhesion. Like right now we discussed, right, focal adhesion, cadherin in the previous lectures, right? So that will be taken in detail, and then comes the cell cycle and cancer. Okay, so we'll try to finish by this week all the 3 topics. Let's meet tomorrow then at 7.